Okay, so let's get right into it. Today, we're going to break down a really cool and super important technique that scientists use to hunt for new antiviral drugs. We're talking about how to stop a virus before it can even make its first move.
This right here is the big question, isn't it? I mean, a virus is pretty useless until it gets inside one of our cells and starts taking over. So, what if we could just slam the door shut and lock it before it ever gets a foot inside? Well, that's exactly what this whole procedure, the attachment assay, is designed to figure out.
To make this whole thing really easy to understand, let's use a little analogy. Just imagine that your body cell is like a high-security apartment, and the virus, well, that's our unwanted visitor trying to break in. Viral attachment is that split second when the visitor grabs the doorknob. If we can stop that from happening, they're stuck outside, end of story.
All right, so to run this experiment and see if we can stop our unwanted visitor, we need a few key players on the field. Let's get to know them.
So first up, we have our Vero cells. These are going to be the apartments that we're trying to protect. Then, of course, we need our unwanted visitor, the virus itself. And finally, the potential hero of our story, a plant extract. We're hoping this extract will act like a high-tech security system and keep that visitor out.
Now for the fun part. It's time to actually run the test and put our new security system through its paces. Let's see if it can do the job.
The setup starts pretty simply. First, you lay down a nice, healthy layer of cells in a dish. That's our apartment complex. Next, and this part is absolutely crucial, you take the virus and you mix it together with our potential drug, the plant extract. You're basically giving the security system a chance to find and neutralize the visitor before they even get to the front door. Then, you add that whole mix right onto the cells.
And this gives you a great visual of the whole process. You see the cells getting prepared, the mixture going in, and all the following steps. But did you notice that little detail in step 3? It mentions incubating at a really cold temperature.
And you should definitely be asking that question. Why on earth would you make everything cold? It seems kind of counterintuitive, right? Well, this is actually the most clever part of the entire experiment.
You see, that cold temperature is like hitting a giant pause button on the whole infection process. It gets just cold enough that the virus can physically latch onto the cell. It can grab the doorknob, but it's too cold for it to actually turn the knob and get inside. This is genius because it lets us isolate and study that one specific moment of attachment. It's exactly what we want to measure.
Okay, so we've hit pause, and we've given our security system a chance to do its thing. Now we need to hit play and actually measure the results. It's time to see what damage was done, or hopefully, what damage was prevented. So what happens next is really important. First, we gently wash everything. This gets rid of any viruses that failed to attach, any visitors who couldn't even grab the doorknob. We only care about the ones who made it that far. Then we add a special gel on top and warm everything up to normal body temperature. This is like hitting play. Any virus that's successfully attached can now get in and start spreading. After a day or so, we come back and count the aftermath.
Now, you might be thinking, hold on, what's up with that gel layer? Methylcellulose. It seems like a kind of random, gooey step to add in there. What's it even for?
Well, that gel is basically a containment field. Think of it like a fence. It keeps any new viruses that get made from just floating away and starting new infections all over the place. Instead, they're trapped, they can only infect the cells that are their immediate neighbors, and this forced, localized spread creates these little circular zones of dead cells that we call plaques. They're visible, and most importantly, they're countable.
So the experiment has been run, the infections have been contained. Now comes the moment of truth. We get to read the results and find out if our plant extract, our security system, actually worked.
So, just like the diagram shows, that final step is all about the analysis. We add a special stain to the dish that makes the living cells turn a color, while the dead cells in the plaque stay clear. This makes counting them super easy. And each plaque we count represents one single virus that successfully attached in the very beginning.
And this is what it all boils down to. You have your control dish, where there was no security system, the unwanted visitor got in, no problem, and you see a ton of plaques. But then you look at your test dish. If that plant extract worked, if it blocked attachment, you see a huge difference. You see very, very few plaques. It's such a clear, visual answer to our question.
So what does a really fantastic result look like? It looks like this. A number like 95% is incredible. It means our security system, that plant extract, managed to stop 95% of the viruses from ever attaching to the cells. In scientific terms, this is what we call attachment inhibition. It's a direct measurement of how effective our drug was at blocking that very first step. So let's just zoom out for a second and appreciate the big picture here. Why is this particular experiment such a powerful and, honestly, elegant tool for discovering new medicines?
The logic is just beautiful in its simplicity. You prepare the apartments, you run the security test in the cold to isolate just the attachment step. Then you contain any break-ins so that you can easily measure them. And finally, you count the plaques to get your final security report. It gives you a clear, direct yes or no answer.
And there you have it, the viral attachment assay. But it's so important to remember that finding a compound that works this well in a dish is really just the beginning of a very long journey. The next and much bigger challenge is figuring out how to turn that promising lab result into a drug that's actually safe and effective for people. So what do you think some of those next major hurdles might be? That's a huge question, and it's the next chapter in the story of drug discovery.